we are ready to start our next senior seminar talk. For those of you that are new, you are welcome to come to the chemistry picnic, which is May 23rd, since you're affiliated with the chemistry department. If you're interested, please sign up downstairs, just outside the elevator. There's a stock room and there's a sign-up sheet, so you can start there. Also, we are going to have another 11 talks, and so that our next one is going to be about HIV inhibition, and that will be here at 4:30 tomorrow. And then at 4 o'clock on Friday, we also have one about moths. At any rate, also want to remind you that you can watch these in perpetuity on YouTube if you go and Google Lawrence Senior Experience in Chemistry. It's my distinct pleasure to introduce Michael. And so that Michael holds a special place in my heart because he started with me doing research. I had started a brand new project working on yeast, and that I had no idea what I was doing. But it was kind of the Tom Sawyer effect, where it's like, hey, look, I'm painting a white fence. Anybody want to help? And I had all these students come in, and Michael came in as a freshman. And so I've always advocated trying to place freshmen doing research that very first year. We usually don't have enough spots and time to do that, but Michael, I'm not even sure how you found me, to be perfectly honest. No, but it's totally cool, right? It's word of mouth, things like that. And what's important is that we had this cluster of 5 people that worked really well together. Everybody had a certain voice and was contributing to the project, and Michael happened to be the detail-oriented one. Worrying about, are we doing that right? Have we made sure that this is done correctly? Are you really sure about it? And it's like, and that's really very helpful because you've got various people doing various, providing to the group various, how should we say, talents. And so he kept the group tight and straight, making sure stuff is happening. If they weren't sure, I was gone teaching in the Netherlands for 3 weeks. People were like, oh my God. And I was like, just email them now. That's a good idea. But then Michael moved on. This is once again really cool. So he explored a lot of different things. He worked in biophysics for a summer. He's going to be and that was here at Lawrence. And then he's going to be telling you about his work at UChicago. And that he's going to be continuing on to get his PhD in technically biochemistry, molecular biology. It's interdisciplinary. It's interdisciplinary. Right, which is perfect. It's a little bit of everything, which is kind of what we always love. It's just a little bit of everything. So anyhow, without further ado, I give you Michael.
Thank you for coming, everyone. So the topic of my title is Characterizing the Ability of Yeast Protein AP3 to Undergo Protein Liquid-Liquid Phase Separation. And I'd like to start off right away with talking about why we should care about phase separation, and that's because it's implicated in disease, and specifically protein misfolding disorders. And a lot of times this is associated with aging, and so as you get older, your proteome will have an imbalance, and your proteome is just the protein content inside your cells. And what can happen is, when you have an imbalance in your proteome, that can cause the proteins to misfold. And this often happens in neurodegeneration. So for things like Alzheimer's disease, Parkinson's disease, and also chronic wasting disease, which we have here in Wisconsin, which is a protein misfolding disorder that can actually be infectious and can be transferred between deer. And what really this is the formation of higher order structures, so it's proteins aggregating together. And so therapies for treating diseases in neurodegeneration can look to deaggregate these proteins, which can be toxic and cause malfunctions in the cell. The composition of phase-separated liquid droplets are protein, as you can see in the purple, RNA, which is the little squiggly lines, and these come together to form these clusters that look like droplets inside the cell, and they lack a fixed stoichiometry. So a great way to think about phase separation in biology is like oil and water, and these are fluorescent proteins that are clustering in vitro inside a test tube, and so they settle into these characteristic spherical droplets. And so, extending that analogy, you've probably witnessed this when you toss salad dressing and seen olive oil droplets form in the vinegar. And what is occurring is a hydrophobic-hydrophilic interaction where the oil is hydrophobic and it wants to aggregate with itself, separate from the hydrophilic environment. And inside the cell, there's cytosol, which is an aqueous or hydrophilic environment. And the cell is membrane bound. And how we think of the cells is compartmentalizing its components is through aggregating their guts basically into these membrane-bound organelles or compartments. For example, the nucleus holds your DNA. A series of structures called the Golgi stack are membrane-bound structures that package and process proteins. The mitochondrion is a double membrane structure. And then, for example, the lysosome is another membrane-bound structure that has an acidic environment that's important for its enzymatic reactions. And so much like we have multiple rooms in our houses that have different functions, we have different compartments in our cells that also have different functions. And this is the stereotypical picture of a cell, membrane-bound compartments, but phase separation is a new way to think about compartmentalization in cells that's not membrane-bound, that has its own advantages. So why does phase separation exist, or how is it functional inside a cell? It's adaptive, and the first way we think it's adaptive is through regulating cell homeostasis. And cell homeostasis is just the normal functioning of a cell in steady state normal in a normal environment. However, and perhaps more interesting is the implication of phase separation in stress and the stress response in a cell. So if you look at the top right image, you can see these proteins that haven't aggregated, but when you have a stressor like temperature shown here, that can cause the proteins to aggregate, as you can see right here. And this is often observed to be required for cells to actually live and survive upon a stressful event, such as temperature. And the fact that these phase-separated liquids don't have membranes, that allows them to respond really quickly and directly to environmental input. The initiation of phase separation is through a seeding event. And so you have these proteins that can bind to a molecule like RNA, shown here, and that can begin to phase separate your protein components. And this can occur even more with continued binding to form hydrogel, so liquid to hydrogel transition, and hydrogels are just a little bit more solid-like. And a lot of time in neurodegeneration, you form even more solid-like structures that can be toxic to the cell. And so what I'd like to get away from this slide is just that there is a gradient of aggregation going from more liquid-like to more solid-like states, and we observe these in vivo in the cell. Concentration is known to be an important factor for phase separation. And so proteins that are involved in phase separation can actually be up to 100 times concentrated relative to the cytosol or the inside of the cell. And how this occurs is through reaching a saturation concentration. As you can see on the right, once you reach this concentration, you have enough protein to drive condensation, and this is how proteins aggregate. However, as you'll notice, there is a double arrow going in two different ways, and what that means is that there's a dynamic equilibrium of exchange. So once you're a protein and you go into this state, you can still go back and things are being exchanged between the liquid-like aggregate and the aqueous phase inside the cell. Multivalency is another property deemed to be important for phase separation. And how we describe multivalency is through containing multiple binding domain partners. And so if you look at the top right, we have multiple red boxes that are binding to the multiple blue circles shown here. So these are binding partners. And when you have multiple of these, it can drive phase separation. So if you look at the bottom left in this diagram, on the x-axis here, we have the concentration of the red boxes, and on the y-axis, we have the concentration of the blue circles, and these are binding domains. And the blue dots indicate that phase separation was not observed. And so this, as you can see in the subscript here, was 3 copies. And so no phase separation was occurred with 3 copies, but when we add a copy, whereas phase separation didn't show up for 200 micromolar of either, you begin to see phase separation by adding just one more binding domain, as you can see I've illustrated up here. And when you add even one more domain, which I did not illustrate at the top right, so having 5 copies of each, phase separation occurs even more readily, because you can see the red dots indicating phase separation occur at even lower concentrations than they did before. The type of binding interactions that are deemed also to be important for phase separation are unstructured regions, also called intrinsically disordered regions. And what these are just regions that lack a fixed conformation. And so if you look at this GIF over here, you'll see right here we have a region that's conformationally flexible, and so it's folding in different ways and it's dynamic, compared to the red or the blue shown here that don't change their fold as much as this region. So this is the unstructured region I'm talking about. Within unstructured regions, we have low complexity regions that are just unstructured regions that contain biased amino acid content that appear important to phase separation. And these are what facilitate unique binding interactions in phase separation. And so proteins that are known to phase separate and form droplets have a higher proportion of these regions than proteins that don't. So all of these unique properties of phase separation come together for an important purpose in the cell, and that's stress. And so, for example, temperature and pH are two stresses that the cell needs to respond to in order to survive. And some studies have made phase diagrams for these proteins that phase separate. For example, this one shown here, temperature on the X and pH on the Y. And this forms a phase boundary indicated by the solid line, where if you cross from one to the other, you can go from the soluble state to the demixed or the aggregated phase separated state. And so, for example, if you're at a pH of 6 and you increase the temperature enough, you can dry proteins to aggregate. So how do we study phase separation and how do we know about it? Well, a great way is to use yeast, which I love. And so, yeast is a great model organism because you can manipulate its genome really readily, and also it grows really quickly. And so it's a great way to study phenomena, cellular phenomena. So zooming in, yeast contain all the cellular compartments that I talked about at the beginning of the talk. And I'd like to focus on the Golgi, as you'll see here, because this is where my research lied. So a model we have for the Golgi stack, which is implicated in the secretory system in the yeast cell, works as follows. So proteins are synthesized in the ER, shown up here, and then they're packaged in vesicles and driven to the Golgi stack, where they go from stack to stack and are altered chemically for different PTMs or different post-translational modifications, and then they can be packaged to the vesicles and go to different places in the cell. And what's interesting about the Golgi stack is that they're not stable compartments, they're highly dynamic inside the cell. And so a given Golgi stack up here in the early state will actually mature and change its molecular composition, and then those proteins will be recycled in a cyclic formation. And so Golgi are really dynamic and a really interesting organelle to study. The protein in the secretory system that I was interested in studying is AP3, an adapter complex that works at the late stage of the secretory system in yeast, and you can see it down here in red. And what it does is it takes protein that has been transferred to the TGN, otherwise known as the late Golgi, and it packages it into, it transports cargo in vesicles to different areas. So if we zoom in here even farther, we'll again see the late Golgi shown up here, and then AP3 concentrates on these vesicles, packages cargo, and fuses with the vacuolar membrane shown here through interaction with the Hobbs complex, and then this allows transport of protein to the vacuole, which is also called the lysosome in other organisms. And this is a great image of AP3, which is a protein complex, which means that it has multiple subunits, and the ones that are most pertinent to this study are APL6 shown in pink and APL5 shown in this purple color. But how do we study proteins like AP3 or any protein in the cell, and how do we visualize it? We have to tag it with the fluorescent protein. So this is not AP3, but it's just an example of a protein's 3D structure. And if we add a fluorescent protein like GFP and we shine a laser on it, it can emit light and fluoresce, which we can detect in a microscope, and which we can then see. However, usually, at least in the lab I was in, we tag them with a ton of fluorescent protein that allows them to be visualized even more readily. This is what a microscope looks like, specifically a confocal microscope. And so you would put your slide here, and then you would shine a laser on it, and that would cause the proteins to fluoresce, and then an image would show up here that you can then do more data processing with. The type of confocal microscopy that I was most interested in was 4D confocal, which is a really powerful technique because you're looking at cells throughout time. So that's the fourth dimension. And the other 3 dimensions are the 3 spatial dimensions. So if you look at the top up here, you have an x and y coordinate, and you can obtain data on one x and y focal plane, otherwise known as a slice. And then you can keep looking at slices until you have obtained visual data for the entire cell, which is really important because if you're only looking at one focal plane, then that's not necessarily interesting because organelles can span multiple focal planes. And what this looks like is optical sections shown down here. And so one organelle might be in one focal plane and another might be in a different focal plane. And so we take all of this 3D data, that's these stacks, and we squish it into a 2D projection. So although this is 2D, you've taken all of that 3D data and put it into a really easy form to visualize. Still, this is pretty busy because cells contain a lot of compartments, as I've talked about before. And so what we usually do when we're analyzing in the lab that I was in is we isolate an organelle of interest, a compartment of interest, erasing the other ones so that we can more easily see it. And what this looks like when you're actually doing it is it's a pretty time-intensive process because you have to make sure that it's properly, even though software helps you, you have to make sure it's properly done for each focal plane. And so, for example, if I was interested in this organelle, then I would have to select it manually with help of software, deleting the other organelles. And I wanted to point this out because it shows that organelles can span multiple focal planes. Returning to the secretory system, if you recall, Golgi matured, and so they changed their molecular composition. And 4D confocal was one of the methods used to actually witness this. And so I wanted to give this example of how it's a really powerful tool. So if you look in the middle panel right here, you'll see that we have isolated a compartment and are following it throughout time. And as you'll see, a compartment that contains a protein tagged with a green tag changes to containing a protein that is tagged with a red tag. So going from VRG4 to SEC7. And you can quantify this with a plot shown down here, so you can see, even though I think it's pretty easy to see looking with your eye, that we've gone from a green signal to a red signal, indicating the molecular composition of the compartment has changed, and that the Golgi matured. This tool and a similar experiment was done to actually form the initial observations that led to my research. And so we have AP3 tagged in green and SEC7 tagged in red, and SEC7 is a late Golgi marker, so the TGN, which is where AP3 transports cargo from. And so throughout time we see AP3 show up, and then it aggregates around SEC7. And you can see the red signal and then the green signal, which was really odd because when similar studies are done with other adapter complexes, the signals actually overlap. And so with green and red, that would be a yellow signal, but you can clearly see here that there's 2 separate signals that are associated but not co-localizing or overlapping. And what this could have meant is that AP3 was actually in a different tubular part, a different part of the membrane of the TGN. And so what this experimenter did, Casey Day, in the lab I worked in, is she looked at endocytosis of the bulk membrane dye FM464. And FM464 will be visualized in red in these slides. So FM464 intercalates into membranes, and in yeast, when you endocytose, it all goes to the TGN, or the late Golgi. And so if you look up here in the top panel, SEC7 is the marker for the TGN, and FM464 was endocytosed because you see these punctate structures, or spherical structures. And you'll see there's yellow. And so FM464 in this study, which was in red, was co-localizing with SEC7, which was in green, or GFP, indicating that SEC7 is membranous because FM464 labels membranes, which makes sense because we already knew that. But then when you look at APL6, which is a subunit of AP3, tagged with GFP, and you do the same experiment, you see that there is not co-localization. So you see red structures and you see green structures that don't overlap, which indicates but doesn't prove that these AP3 aggregates are non-membranous. And so these two experiments are what led to the hypothesis that AP3 phase separates inside the yeast cell. The first experimental strategy was to use confocal microscopy to look at special strains of AP3. And these strains were created by Leanne Blodgett in the lab that I worked in. And what was really interesting about one of the subunits of AP3, APL5, is that it contains predicted unstructured regions through special bioinformatic techniques. And so there was two predicted unstructured regions. There was one that was shorter and one that was larger, and they just overlap. But they are both in the C terminus, or the end of the APL5, AP3 subunit. And so what she did is she took these unstructured regions and she truncated them, leaving the first 849 with the first 634 amino acids. And what was important about these strains is that if AP3 aggregates and you no longer have unstructured regions, we want to see if it messes up its normal localization and aggregation in the cell. So I repeated the experiment of looking at AP3 tagged with SUC7. Wild type, or WT, just indicates that this is a normal cell, other than the fact that we tagged them with fluorescent proteins. And as you'll see, AP in the bottom panel in the isolated compartments, depending on what AP3 is, you'll see that AP3 aggregates around SUC7 in red, but doesn't colocalize, which is what we observed before. And when we do this with the shorter truncation, we see that there is a similar result. However, using epifluorescence and looking at the larger truncation, we see a really drastic phenotype. So here's wild type with the AP3 puncta, or spherical structures. But then in the larger truncation, the signal went completely diffuse, which is really interesting because that is the first evidence suggesting that these unstructured regions, which are known to be important for phase separation, are important for AP3 aggregation. And it's also cool because there seems to be sort of a gradient going from the shorter truncation to the larger truncation. So the shorter truncation still had some diffusion of the AP3 signal. The next thing I looked at doing was performing a functional Western blot assay. And so, yeah, we've chopped off part of this protein and visualized it, but perhaps we just chopped off so much that there's no AP3 on the membrane anymore. And if you'll recall, AP3 transports cargo from the late Golgi to the vacuole inside yeast. And so what my assay was doing, the functional assay, was to assess AP3's ability to transport cargo in these strains that contain the diffuse phenotype. And what this involves is collecting different fractions that isolate different organelles. And how you isolate fractions in cell biology is you use differential centrifugation, which involves a centrifuge shown here. And inside the centrifuge is this great little structure, this great little compartment that you can put your samples in. And then when you spin this structure, you can actually create different fractions, such as a supernatant and a pellet, that will separate different cellular guts or different cellular components based on speed. And so what this looks like is you have your initial cellular guts. You can spin at a low speed, pelleting larger cellular structures, and continually to collect the supernatant and putting it into the next tube, spinning it at a little bit of a higher speed, and collecting medium structures, and then eventually collecting all the different fractions you want. And then you can take all of these and then analyze them through, for example, Western blotting. So how Western blotting works is you take these different fractions and you put them in these different wells, subject them to an electric field that will separate them by size. And so you'll see these bands up here that you also have a marker to show what size these proteins that you're isolating actually are and to verify their size. And then you have this gel with a protein in it, and then you can transfer it to a membrane where the protein just sticks on top. And this is the membrane you're actually analyzing. So what this looks like when you're analyzing is you take antibodies and you bind them to the protein. Importantly, antibodies are very specific to the protein, your protein of interest, and so that's why you use them. And then one of the antibodies, or the last antibody you put on, has an enzyme that when you put on a substrate can actually cause light to be emitted, and then you can visualize it. And what this looks like is a Western blot where you have these different bands that you can actually quantify or look at qualitatively depending on your experiment. And so just to make this as clear as possible, this is your membrane and this is your protein from a side view, and then this is a view looking down where this is the protein and this is the membrane. The Western blot assay that I was interested in looking at was developed to assess AP3 function. And how we do this is we isolate fractions that contain, for example, the Golgi membrane, so that's P100, shown here. And then another one that isolates the vacuolar membrane, which is shown here. And just to make this very clear, I put in this repeated picture where we're showing that we're assessing AP3's ability to transport cargo from the late Golgi in this fraction to the vacuolar membrane shown in this fraction. And so these bands correspond to an AP3 cargo, which is alkaline phosphatase. And we know that this assay is sensitive enough to detect AP3 function because if you knock out one of the subunits in AP3, as you can see on the right, our ability to transport to the vacuole, the P13, has been altered. So you'll see this band is lighter, so cargo isn't going to the vacuole. So just to be as clear as possible again, the cargo is shown in this purple triangle, and it's being transported to the vacuole. And by isolating different cellular organelles, we're looking at content of this purple triangle or the cargo in these different fractions. The specific antibody I used in my assay was against Pho8, which is a specific alkaline phosphatase in yeast. And it has a molecular weight of 63 kilodaltons, which, as you can see, is between 75 and 50, shown in this band. In the pilot assay, I was able to show that our antibody was working correctly because between 75 and 50, we're getting bands. And this was important because we wanted to make sure that we're selecting the correct protein. Preliminary results were pretty exciting because when we're assessing AP3's ability to transport cargo from the late Golgi to the vacuoles, from this band to this band, when you look at the larger truncation, which had the completely diffuse green phenotype, so it had this diffuse altered localization in the yeast cell, it still appeared to be able to transport cargo to the vacuole in this band. Because if you look at wild type, there is a heavy band here, and if you look at the larger truncation, there is also a heavy band here. Now what you'd want to do is quantify this, but I only had 10 weeks, so I didn't have time to do that. And you can see differences, for example, in this band, between this band, and this band does look lighter than this band, but still qualitatively it appears the ability of the AP3 to transport cargo remains largely unaltered in the truncation strain shown on the left, which is really interesting because despite the fact that it had that weird localization, still some AP3 was acting on the late Golgi membrane to transport cargo. So we compared the wild type to the larger APL5 truncation, but you'd want to have a negative control, so when AP3 is actually knocked out and not existent in the cell, and you'd want to do that experiment to make sure that your assay is working properly. So I was charged with also making that strain. However, it was really difficult to make because when you knock out genes from yeast, it can alter their normal cell homeostasis and their function, and you replace it with another gene, and so it gets pretty complicated, and it's not always easy to just remove a protein from a cell, even if it's not essential for survival, which AP3 isn't. So what I think is the coolest experimental strategy of my project was attaching the unstructured regions to GFP. And what this would look like is taking your GFP, which again is the fluorescent protein, and adding unstructured regions that were predicted to be, that were shown to be important for normal AP3 aggregation in my previous experiment. And if you add them to GFP and you visualize it in a yeast cell, if that caused GFP to co-localize with AP3 signal, that would again be further evidence that these unstructured regions are important for normal AP3 aggregation. However, when I added both of these DNA constructions into the yeast, the signal went completely diffuse. As you can see, there's just green signal flooded throughout the cell. And so this result really isn't interesting. It doesn't say that it doesn't offer evidence that the unstructured regions aren't important for AP3 aggregation. It just doesn't say that they are. Because what we could have done is just express too much protein inside the yeast cell, where that's all we're visualizing, and there's too much in there to even see any other structures. I also was charged with creating a strain to study AP3 using a really cool microscopy technique called CLEM, and that stands for Correlated Light and Electron Microscopy. And so a little bit of background on microscopy and resolution. So you have your eye over here, which only can look up to a certain resolution. And that's where light microscopy is really useful, shown here, because you can visualize subcellular content. And specifically why light microscopy is useful, as I've been showing throughout the talk, is that we can use light to specifically tag specific proteins. And so with light microscopy, we have specificity. Still, light and its wavelength have a limit on resolution. So cellular structures and proteins of interest, you want to visualize at an even smaller level. And that's where electron microscopy, shown here, comes into play, where you have a way better resolution because of how electrons propagate. And so with electron microscopy, you can actually see subcellular features with way better resolution, such as these stacked Golgis. And so with CLEM, this is an example image that was obtained, or an example data that was obtained with CLEM, where you have this fluorescent tag that's able to show you specificity. So here, red indicates late Golgi, overlapped with the resolution of electron microscopy, where you're looking at, you can clearly see the tubular structure of the Golgi. And so it's pretty clear, as you can see down here, that light has a resolution limit. And so this is why adding electron microscopy would be really useful. Because if you recall, it hasn't been proven that AP3 is non-membranous, but if we looked in with electron microscopy, we could actually show that these AP3 structures are not on the membrane, which would be further evidence that they're phase separate. In conclusion, the predicted unstructured regions do appear to be important for normal AP3 localization, and even the larger truncation strain, even the larger unstructured region, which when you truncate it has that diffuse phenotype, it can still exhibit AP3 function, which is really interesting. But attaching the unstructured regions to monomeric GFP isn't sufficient to localize GFP to AP3 structures. Future directions are to successfully create the knockout strain to have a negative control for the Western blot assay. And if you recall, multivalency, or having multiple binding domains, is important for phase separation. And so if you looked at a tetrameric protein, which is a protein that has 4 subunits, and you attach the unstructured regions to 4 subunits instead of just 1, then that could perhaps be a better way to assess, to repeat the experiment with the GFP, because if you have 4 unstructured regions, perhaps that would promote it to aggregate with the normal AP3 aggregates. And then also perform CLEM on the optimized halo tag strain that I created, which was like at the end of August, and so I'm sure they've done this, and I'm not really sure what the results are. And if AP3 does phase separate, there should probably be a purpose for it. And so what my experiments were doing were really preliminary to look at different characteristics we know of phase separation, how they might apply to AP3. But once we've gotten that nailed down, we really want to look at how AP3s and its phase separated properties, if it is, are functional in a cell. And as if you recall, environmental stressors and environmental conditions are important for this. And so future work would be to look at how AP3 actually responds to different stressors and how it might respond to, how it might be important for normal homeostasis. And what's interesting about AP3 is that it's actually in these it's actually in these aggregates under normal cell conditions. So perhaps it's different from other phase-separated liquids where they get released upon stress. I'd like to thank everyone in the Glick lab for helping me with these experiments, specifically Ben Glick, who is my PI, and then Jason and Natalie, who were the grad student and PI that helped me, all the RU coordinators, and then the whole RU program for the opportunity. And then everyone at Lawrence University who's helped me along the way, such as my advisors Dave Hall and Austin Fleshman, and also Daniel Martin in the stockroom for all of his help, and of course all of my peers who have showed up today, who've been very supportive. And any questions?
Yeah. How would you quantify the differences after knocking out the IDP?
In the Western blot? Yeah, so what you would do is you would upload this image to something like image processing software like ImageJ, and then you would actually select a band, and then based on like the intensity of the signal compared to a standard protein concentration, you would be able to have a software based on the intensity, based on relative to the intensity, show you how much protein is actually in those different fractions. Yeah, but it would have taken a lot of time to actually do that with this assay. Yeah.
So you said that on one of the parts you put the GFP fluorescent marker on. You said you added a lot more than the typical amount. So you had like multiple GFPs per.
Oh yeah.
So is the signal that you're getting then, was that because you didn't have a high enough concentration of GFP to detect it, or was that because? So again, I'm curious what the reasoning was to have so many.
Yeah, so you don't have to have that many. A lot of studies only have one, and it just depends on what you're actually looking at. This lab is really always has like 6 copies or more, like not more, like 6 copies of fluorescent proteins tagged to the proteins of interest. Which, when I first arrived, I was like, this is kind of crazy because how is that not affecting function, especially when you're transporting cargo? But honestly, like there are ways to check that, like through like assays like the Western blot assay. But the reason why you want to do that is because it just increases signal really well, and especially because yeast are so small and these compartments are so small that you really want to get as good of a signal as possible to like quantify these different compartments overlaps and their different dynamics. And when you add more fluorescent protein, it also helps with things like photobleaching.
To follow up on her question, is the green fluorescent protein by itself tends to aggregate. So it forms dimers and trimers and stuff. And so was that all a concern experimentally?
Yeah, so that's a good point. I didn't even, like, when I was researching, I always just thought of it as like these tails of proteins that were like following the protein of interest. And do you know like if it's one type of GFP, like monomeric GFP, or
there's all kinds. So
yeah, so I don't know which. Yeah. I don't know if all types of GFP aggregate, but that would be an interesting thing to consider, like how would aggregation of GFP maybe even be affecting like aggregation of AP3 when you're looking at it. I don't know, but yeah, that's interesting.
Other questions? Oh, good. I've been saving all of mine. I've heard like 3. So. Speculate on what AP3, why it would be a phase separation. So I mean, for example, you have proteins that are transcription factors that are very well-known aggregates, and it makes sense because if it aggregates, it doesn't function. If it deaggregates, it functions.
Yeah.
Same thing with translation factors. If it aggregates, it doesn't function, etc. Why would this make it?
Yeah. So I have like can I draw on the board?
Sure.
Okay. So. I have, like, so if this is like your Sec7, your late Golgi, and this is your vacuole, and AP3 transports stuff to there, what we see is you have some AP3. The idea before all this research was that you have AP3 on the membrane and it's just moving stuff this way. But what we see, if you recall, is like this type of thing where it looks like there's like a pool of AP3. And so perhaps what my hypothesis is that this probably responds to different stressors. And when it responds to stressors, you take this reservoir and you maybe release it onto the membrane, something like that maybe, and that would give you more AP3 to transport more stuff to the vacuole, which I don't know why you want to do that, but that's, I think that might be one of the reasons. I know since AP3 transports stuff from the late Golgi, which is the late Golgi is where endocytosed cargo comes, perhaps like stress damage it or like causes protein misfolding or something on the plasma membrane, and that needs to be degraded at the vacuole, and so you'd want to like shuttle a ton of it to there, but honestly, I don't know yet. I mean, it's more research is going to be required.
Other questions? Okay, just one more. Why did you? I'm sorry, but what? So, and the one thing the audience doesn't fully appreciate is that the old-fashioned way to refer to this is as prions.
Yeah.
So, particularly in the yeast model system, why it appears your lab started studying Golgi transport and then chose to call it phase separation instead of saying AP3 is a prion and we are studying prions. Do you know why they went that direction?
Yeah, so I think, so prion is like, that term is heavily associated with like neurodegeneration, and I think what's interesting about specifically phase separation is that it's really interdisciplinary, so people study it, like people model it, like they don't even look at it in vivo, and then also people will study it like up from its like biological function to like chemically and physically like what's actually going on. And so I think like the nomenclature, there was like someone who was like, oh, didn't know anything about prions and was like, oh, this is like reminds me of phase separation, like oil and water, and so we'll call it that. But I think phase separation probably was first applied to cytoplasmic proteins, not in the nucleus. And then people probably realized, hey, this is like other aggregation. And then so like, then all the terms got kind of confusing. But I think that, yeah, it just depends on what you want to call it. But yeah, it is interesting. There's so many different ways to call it. Yeah.
Yep.
Do someone?
Oh yeah? Okay, let's take a